Okay, I just thought I'd go live today. If there is anyone out there that wants to do some practice for the biology, um, yeah, put up some questions on the chat. You need to subscribe before you can put questions up, and you can ask me any questions to do with the biology papers this week. There's a biology A level paper today, and there's biology GCSE papers this week, paper 2 on Friday. So feel free to ask any questions you like, biology A level or GCSE. So if you're revising, so if you're revising, so I've got a biology GCSE question here. So can go through this first of all. So what we have here is one of the topics that's coming up in the biology GCSE. On Friday, you've got inheritance and variation, all about genetics. Let's go through this question. So, looking at this question, which two statements are true for sexual reproduction in humans? Gametes are formed, offspring are clones, offspring are genetically identical to parents, only one parent is involved, the sperm and egg fuse. So, let's have a little look. Are gametes formed in sexual reproduction? Are offsprings clones? Well, we know that offsprings in sexual reproduction aren't clones. You look different than your brothers and sisters, you're actually not a clone. Offsprings, therefore, are not genetically identical to the parents. During sexual reproduction, which is how we are produced, we don't look like our parents. Well, we don't look exactly like our parents. Is there only one parent involved in making you? Well, in sexual reproduction, there has to be two parents involved. One that contributes the male gamete and one that contributes the female gamete, i.e. sperm and egg. So we know this is wrong. And this leaves our options as the sperm and egg fuse in a process known as fertilization, and also gametes are formed. Before sexual reproduction can happen, sperm is made in the testes and eggs in the ovaries. Obviously in a plant it's slightly different. There's pollen produced in the anther, but plants also have eggs in an ovary. So these are the two correct answers here. Now, you also need to know what is the probability of a mother and father having a boy or a girl, i.e. having a male or female. And we can represent that by this Punnett square, which we show down here. So this is a Punnett square, and we can see that we have XX written here. Now these X's represent the chromosomes, the 23rd chromosome in humans, which is known as the sex chromosome, the one that determines whether you are a male or female. Now a mother contains an X and X, two X chromosomes. She gets one of the X's from her mother and one from her father, so she inherits those two X's, and that's what decides that she was a mother or a woman. And the father has an X chromosome, but the father doesn't inherit an X and an X. The father inherits an X from the mother, but can inherit a Y. So boys or males have the chromosomes, the sex chromosome combination of X and Y. Now what we can do is we can combine these. So when men make their gametes, they make sperm, which have tails. You don't have to draw that. That's just to show you what's going on here. And you can see one has been filled out for us. The probability there, we've got one there is XX. If this X sperm meets that X egg, we're going to get another XX. And if this Y sperm meets that X, we're going to get an XY. And if this Y sperm meets that X, we're going to get an XY. So the probability is 50%. Now I'd like to highlight this. If I'm in an exam, I always want to highlight what I've written here and state the phenotype. So this is male, and the probability of having a male or having a female is 50%. You can put it as a percentage or as a decimal like that, 0.5, or you can say 1 in 2 or half. So there's lots of different ways to represent your answer there. Okay, let's look at another question here. So this other question we've got here is draw one line from each hormone to the change the hormone causes at puberty. And one of the topics that's coming up in biology paper 2 on Friday is the endocrine system and homeostasis. So how we control our internal conditions and the role of hormones in the body. So estrogen has many roles in the body, and here we've got changes at puberty that estrogen is in charge of. Well, estrogen, is it found in a boy or girl? Well, estrogen is mainly found in women. It's made in the ovaries, and it's in charge of secondary sexual characteristics, things that physical changes that happen in women when they go through puberty. So what we're really looking at is estrogen is going to help with breast development, because eventually women are going to produce milk to feed babies, so their breasts need to develop. They need to develop the tissues that do that. Their hips need to widen to make childbirth easier, and of course, like boys, they get pubic hair on the genitals or hair under the arms. Now in boys, what is testosterone in charge of? Does it make your skin turn lighter, your voice become deeper, or wisdom teeth appear? Well, it does a lot of the things that estrogen does, like pubic hair in the genitals, hair under the arms, but it does some other things. It widens the shoulders of boys, and one of the other things, it makes the voice deeper. So if you got boys going from year 9 to year 10, you'll notice in your school their voices start going all over the place while they make that change from the testosterone. Now, we've also got here something else that's coming up on Friday, is the control of fertility. So the control of fertility, condoms, what are they used for? They prevent the sperm reaching the egg, and the oral contraceptive pill, okay, that stops the egg maturing. So the oral contraceptive pill tends to contain estrogen, and one of estrogen's roles is to inhibit follicle stimulating hormone, which stops the egg maturing. The pill can also contain progesterone, which is another female hormone that also inhibits FSH. Glad to hear, Nihat, that your exam went really well, so well done. If you've got any questions for biology paper 2 and you're doing IGCSE, I see, if you've got paper 2 coming up and you're not doing combined science, but maybe you're doing combined science and you've already finished your exams, because if you're doing combined, you'll have finished with IGCSE, because you just do paper 1 in IGCSE combined. If you're doing separate science, you can ask questions about the biology paper 2. So feel free to ask any questions you need to. So got another question here, which is on DNA. This could be potentially coming up in your biology paper 2. So in which part of the animal cell is DNA found? Yep, I can do the adaptations of the lungs, definitely. So, in fact, as you've asked that question, let's zing to that straight away. So, adaptations of the... Yeah, you're right. I'm just going to go through, as you've asked the question. And there's not many people on here yet. So, as this is just a last minute random. On Thursday night, I'm going to be doing, at 8 o'clock on Thursday, I'm going to be doing a last minute, 2-hour revision for the... biology paper 2, where I'll take any questions and go through lots of exam questions. Okay, so with your lungs, you've got the structure of the lungs, where you have trachea, then you have the bronchi, and then these bronchi, if you imagine like an upside down tree, they split. I'm not going to do too many branches because it'll just take me forever. They split into tiny tubes, and these tiny tubes are known as bronchioles. Now the bronchioles don't contain cartilage to protect them because they're inside your lungs, but your trachea and your bronchi, the two big tubes, one to each lung, contain cartilage to give them a bit of protection. Now your air comes in here, we can't filter the air to just take oxygen. We take the whole mixture, the argon, the oxygen, the nitrogen, the carbon dioxide, whatever's floating about in the air, we breathe in. Now, how are the lungs adapted? Well, the first adaptation of the lungs, so adaptation number one, lungs contain millions of alveoli. And why is that important? How does containing millions of alveoli help? What do you think it does by having so many of the alveoli? Alveoli are highly folded sacs. It gives the lungs a large surface area. So that's the first step. If you have a large surface area, you're going to do rapid diffusion or gas exchange. So the fact that they've got millions of alveoli, and then we need to say the alveoli walls are, you can say 1 cell thick, or you can just say are thin. Okay, how does that help? That's just a description that they're thin, but how does that help with the function, which is gas exchange? So that helps by creating a short diffusion route or pathway or distance. So you can say a short distance for gases to move. The kind of term for that is short diffusion pathway, or you can say short diffusion distance. So having thin walls. Good blood supply. How does good blood supply help? All exchange surfaces in animals normally have, or certainly in mammals and fish and birds, have a good blood supply. This maintains the concentration gradient. What you'll find there is that if you zoomed in on an alveoli, like if you zoomed in on one of these and you went and zoomed in, not only are they highly folded, which gives a large surface area to each of the millions of alveoli, but they have a blood vessel wrapped around them, which is a capillary, and that means that it's a very short distance for the oxygen to cross into the blood and the carbon dioxide to go back the other way. So it happens quickly. The blood doesn't have to stop like a tube train, open the doors. It can just be moving past and things can just be zipping it on and off. So your three main things are large surface area from the folding and the fact they've got millions, thin walls, short diffusion pathway, and good blood supply maintains the concentration gradient. There's one more bonus one for the lungs, and that is that the alveoli surfaces are moist, and that allows gases to diffuse more easily. So the alveoli surfaces are moist, gases diffuse more easily with a moist surface. So there's like a liquid on the lungs there. Oh, I've kind of meshed all that up. Cool. Yeah, short diffusion distance. It increases the concentration gradient. Blood capillaries increase contact area with alveoli diffusing. Yep, you've got it. So you've really got the adaptations of the lungs there. Now, for ventilation is something different. So for ventilation is where our lungs move up and down in order to ventilate the lungs. How do we bring air in? It's not diffusion. Air does not diffuse into my lungs. So how does air get into your lungs? How can air possibly get into your lungs? Well, the way air gets into the lungs is we create a big volume. We lower the pressure so that the pressure in the lungs is lower than the external atmospheric pressure, and then the air moves in down a pressure gradient. So in order to do that, our ribs move up. So you can put your hands on your chest, and if you breathe in, your ribs move up. Your diaphragm moves down or flattens. This creates a big volume. Big volume means the particles are far apart, so a low pressure air rushes in. So although you can't tell the difference, it happens so quickly, your ribs move up before the air goes in. So when you breathe in, it happens so quickly you can't tell, but your ribs move up and that causes the air to go in. You create a large volume, low pressure, and the opposite is ribs move in, diaphragm moves up, small volume creates a high pressure and the air moves out, and that's ventilation. That's right. Your diaphragm muscles contract, external intercostal muscles contract, pulling the lungs outwards. You've got it. Diaphragm flattens. Yeah. You can say it moves down or flattens, and instead of moving up, you should technically say the diaphragm domes, like it forms a dome, but because it never really goes down that way, but it flattens like that. But technically, you could describe that as moving downwards. But yeah, good. Yeah, muscles always contract, so good. You've got it. So you've really got the adaptations of the lungs down. Are there any other parts of your GCSE you want to go through? Sorry. So you could be asked, oh yeah, be prepared. If I showed you this and said, this is the alveoli of a smoker, why do you think smokers, or it could be emphysema or a lung disease, why is this alveoli less efficient at gas exchange? Yeah, I'll go through DNA. Why do you think this alveoli is not efficient at gas exchange? Yeah, the walls. So depending on what you've got, if you've got emphysema, the walls break down. They're not such a large surface area. So you'd say, yeah, disrupted surface area. You can say that there's scar tissue, which creates a bigger diffusion pathway. Yeah, so surface area decreases, rate of diffusion decreases. You've got it, which means you get less oxygen. How's it going to make you feel? If you have less oxygen, less respiration, less energy released, you're going to be tired and fatigued. Also, if you scar your lungs, you'll get a larger diffusion pathway. You get scars all over the lungs. Cool, right. Let's zing to... So DNA... In which part of the animal cell is DNA found? You said earlier that was the nucleus, and you are spot on. It is the nucleus. Letters A, C, G, and T in the diagram represent 4 different compounds called... Do you know what those letters represent? A, C, G, and T? Very good. Nitrogenous bases. Yeah, so you can just call them bases in IGCSE, but they are nitrogenous. One strand of DNA in section X contains the following sequence of these compounds: TAT, GGG, TCT, TCG. Now, when I get given a strand of DNA, because it's a triplet code, I'm going to split it to make it easier for me to read like that. So I'm going to try and split it so that I don't mess up the reading of it. And it then says 3 bases equals 1 amino acids. So how many amino acids? We've got 4 here. Okay. Now I could ask you, what would be the code? I could ask you, what would be the code of the mRNA made in transcription, but I don't believe that's in your spec. So in that situation, you just write the complementary code. But I don't believe it's in your spec, because in mRNA they have uracil instead of thymine, so it would actually be AUA, the opening to that. But I think that's beyond your spec, so you're cool with that. The section of DNA, let's look here. The section of DNA. One secondo. I see what you're saying. Yeah, my video's over the top. There we go. The section of DNA described in part A3 is a small part of a gene. The sequence A, C, G, and T in the gene is important. Explain why the sequence is important. What does the sequence do? So what is this TAT, GGG, TCT, TCG? What does it code for, and why is that important? Complementary base pairing. You do need to know about complementary base pairing. But here, you could say, yeah, because of complementary base pairing, you'll get specific. But more importantly here is really just to say, yeah, you could say each triplet codes for an amino acid, and the sequence of the amino acid decides which protein is made. Basically, you're just linking that if you had different, you could answer this question in many ways, but if you had a different code, you get different amino acids and a different protein. So imagine now I have an enzyme. Let me just see where I've stuck my video. I've stuck my video right there, so let's move that. My camera right there. So imagine now I had an enzyme. Okay. If you had an enzyme and you got the wrong code, then the active site may not work because the enzymes are proteins. So the code's very important to be exact, and mutations to the code can create changes in the type of protein that's made. Some beneficial, some not important at all. Okay, yeah, that's right. Yeah, wrong protein coded for, that's right, because you get different amino acids. Okay, so something else you probably need to know there. What do we call the structure of DNA? What have I drawn here? That's absolutely right. You get you potentially would get a frame shift. Frame shifts occur with specific mutations called addition or insertion or deletion, but frame shifts don't occur with substitutions, which they can often be silent. Yeah, you got a double helix. And why have I done it in two colors, your double helix? What do we call these individual strands? What are they? What are the individual strands called? Each individual strand is a polymer. Oh, it's a good question. The chromosome is the structure that the DNA is wound into. So I don't have a ball of string, but if I had a ball of string, when it's all wound up into a ball of string, we would... No, I don't have a ball of string. But if we did, we would call the whole thing a chromosome, but it's actually made up of DNA. So both of them together make up the chromosome. Each strand is a polymer made of monomers called nucleotides. So each strand is a polymer made of monomers called nucleotides. I'm just going to see whether you need to know the structure of a nucleotide. Hold on, I've got my book right here. Let me just see for IGCSE. Boom, boom, boom. It's there, 92. You've got to know DNA is a double helix. You have to know about the complementary base pairing. Yeah, you need to know how many possible codes there are. So, yeah, you need to know, for IGCSE, you need to know how many possible codes can you achieve from 4 bases. So can you answer that question? You need to be able to answer this question. How many possible bases, codes can you achieve? Or codons. You can call them triplet codes, or you can call them codons. It's up to you. And the answer is 4 to the power of 3. So you're arranging 4 letters in 3s. So you go 4 times 4 times 4 equals 64. So you need to know, that's no problems. So you need to know, because there's 4 letters but they're arranged in 3s, you do 4 to the power of 3. 4 times 4 times 4 is 64 codons, or 64 codes. They actually call them codons in IGCSE. So a triplet code you call codon. You wouldn't lose the mark by saying a triplet code. It's the same as codon. How many different amino acids are there that they code for? Do you know how many different amino acids there are? So you need to know that as well. I've got my IGCSE syllabus open now. So how many different amino acids are there that they code for? There's 20 amino acids. So if there's 64 codes and only 20 amino acids, what does this tell us? Yeah, one codon for one amino acid, but actually the fact that there's 64 possible codes and only 20 amino acids out there in nature, this means that all the proteins around us are only made up of 20 amino acids, and there's 64 codes. This means some amino acids have more than one code. So those amino acids, sometimes they can mutate and a nucleotide can be changed and it still codes for the exact same amino acid. So for instance, you could have like GGC and GGA both code for the same amino acid. So, and some are called stop codes and some are called start codes. Some codes don't code for an amino acid at all. Yeah, that's right. Methionine, yeah, and cytosine. Yeah. So most of the time with proteins, we say they contain carbon, hydrogen, oxygen, and nitrogen, but some proteins have sulfur in them as well. How are you with transcription and translation? So if I talk you through transcription and translation, transcription occurs in the nucleus. And what happens here is your DNA, so let's say the DNA is like that. Along comes an enzyme. Okay, cool. You're cool with that. So an enzyme comes along and it opens up the DNA where it joins. So it's going to join onto the DNA on a particular strand. Yeah, very good. Yeah, it causes it to unzip. So yes. So which one is the template? In this situation, the template is the green strand I've drawn, and the other strand is not being used at all. So this blue one is the enzyme. Do you know the name of the enzyme? It's called RNA polymerase. So RNA polymerase joins at start of gene. You're absolutely right. You'll get a mark for saying unzips DNA. It breaks the hydrogen bonds between the two strands of the DNA. Absolutely. One strand acts as the template. And this is where you can bring in your C-base pairing, your complementary base pairing. So what we can do is say uses the template to make mRNA by comp base pairing. In the exam you will have to write complementary, but it's a bit long for me. Okay, yeah, in the CGP they mention RNA polymerase, but it might be that you just need to say the word enzyme. So in that case just say enzyme, and I think you said, yeah, so in that case just say an enzyme causes it to unzip. It then uses the template strand to make mRNA by complementary base pairing. What happens to the, let's make our mRNA a different color. So it's going to take nucleotides, and then as it moves down, it joins these together like this, and it makes a strand. Then the strand of mRNA leaves the nucleus. Yeah, that's right. And then mRNA, you just say leaves the nucleus. If you were studying A-level, you say via a nuclear pore, because there's tiny holes in the gaps in this membrane of the nucleus, in the nuclear membrane. Yeah, that's right. Detaches from the DNA and leaves the nucleus. And that is transcription. And translation happens later. So we say translation. Great. Yeah, and there's a little bit... Good. Yeah, translation happens on the ribosome. Do you remember what happens there? The worst ribosome in the history. You're absolutely right. What brings the amino acids to... What is it that brings the amino acids? This is my amino acid. What brings them over? Binds to the ribosome, and then what comes down? And again, by complementary base pairing, what joins together with the mRNA, bringing different amino acids down, depending on the code. Yeah, good. So the tRNA is this. Here is the tRNA. It's like shaped like a leaf or a clover. This is bringing them down here, and it has complementary base pairs here. Do you know what these regions are called? You good. Anticodon to codon. Which one has the anticodon and which one has the codon? So does mRNA have the anticodon or tRNA? You're right, the anticodon binds with the codon by complementary base pairing. And the different amino acids I've got here are like the triangle and the square. Anticodon is found on the tRNA. Yeah, well done. And what will happen is the bond is going to form here, okay, between them. And this tRNA molecule is going to go. So that one's going to bugger off and go away, leaving that one behind. And then the next one comes down and binds, and then that one leaves, and then the next one and leaves. You don't need to know that for IGCSE, but eventually, once they reach something called a stop codon, a code that says stop, you're done now, then the protein will detach, and your protein will be made up of different amino acids. Yeah, a peptide bond forms between the different amino acids. So this tRNA is going to disappear over here, but it won't disappear actually until the next tRNA arrives. So the next tRNA will come down and attach, and you'll end up, so you might end up with your final protein, geez, looking like that, and then that will detach and go off on its way. And that's translation. So you'll be totally cool for translation. Is there any other topics you'd like to look at? Or do you have any questions? Oh, it's a peptide bond rather than a polypeptide bond. Polypeptide describes the protein. The peptides are the tiny bonds in between. Yeah, the start codon is AUG. You won't need to know that. So if you've got it, let's have a little zing through. Okay. Let's have a look at a fermenter. This is going to be the crustiest drawing of a fermenter you have ever seen. Doesn't really matter how they look, he says. So with a fermenter, it's going to have a paddle. Oh my gosh. I don't even know what that is. That's supposed to be a paddle that's going to spin around, okay, and keep our mixture evenly mixed and spread the heat evenly through. You're then going to have a water jacket around the outside, and the water jacket is going to make sure heat comes away. What do we put in a fermenter? What type of organism do we put in a fermenter? It doesn't really matter what it looks like or whether it looks like the one in your book. It doesn't really matter. You just got to get the principles right. So you need to put nutrients in, and what type of microorganism goes in there. Depending on what you're going to do, you're going to use bacteria. If you're making insulin and you've genetically engineered a bacteria, you need to put bacteria in the fermenter. They're going to produce insulin for you for the diabetics. And we can make tons and tons of insulin, which is much better than before we had fermenters. We had to crush up the pancreas of animals and squeeze the insulin out of dead animals and inject that into people. Now we have fermenters. We put the gene for making insulin into bacteria, we bung them in a fermenter, and they reproduce very rapidly because they do asexual reproduction, which is a lot faster, and they produce lots and lots of insulin. So we need a hole, we need a tube going in to dump in our nutrients. Yeah, we can put bacteria or fungi like yeast. If we want to make alcohol, or we want to make something from fungi, or we can do that. There's loads of different products. This is called a water jacket. So this keeps the fermenter from getting too hot. You can go into lots of detail about this. Why don't we want it hot? We don't want to denature the enzymes of the microorganisms. We don't want it... we want them to work at their optimum temperature. So in order to make sure that's happening and that they're working at their optimum temperature, I'm going to need a temperature probe. Now, a temperature probe is essentially a fancy name for a digital thermometer. So that is going to tell me, if my yeast work best at 34 degrees, I want to keep it at 34 degrees. Okay, yeah, that's right. Also, the paddle, where are we? Over here. Our paddle is going to keep the mixture evenly mixed. Paddle keeps nutrients evenly mixed. So all can't see what I'm writing now. So all, there we go, microorganisms, yeah, are supplied. Oh, I could waffle on for ages, but the paddle is going to keep the nutrients mixed, make sure all the microorganisms are supplied. It will help keep the heat and the temperature evenly mixed as well. I'm going to need something for waste gases, otherwise my fermenter will explode. Yeah, distribute heat and nutrients. What waste gases will microorganisms release? So what type of waste gas will your microorganisms release? Depending on the microorganism, but what's the most common one we're going to be releasing? So what's the most common waste gas that living things are giving out? Yeah, you're right. CO2. We need carbon dioxide out. Otherwise, your fermenter could explode. And people do have fermenters explode. I know somebody who tried to make wine at home using yeast. They tried to make alcohol at home and they didn't have something for the exhaust gases, the waste gases, and all of their things blew up. They actually exploded. So, yeah, pressure, explosion. You're right. You may need air in. You may need air in. You might be saying to me, why wouldn't I need air in? In what type of respiration might you be doing where you don't want air to go in? So let's have air going in here. What? Yeah, we need air for aerobic respiration. But let's say I'm making alcohol. I need to get my yeast to do anaerobic respiration. I don't want to give them oxygen. They might not make my alcohol. Because with fermentation, we make ethanol and carbon dioxide. What about if I'm making yogurt in a fermenter? I want to force the bacteria to make lactic acid. And remember, yeah, that's right. So if I've got yogurt in there and I want to force the bacteria to make lactic acid that turns the yogurt sour, so I'm making a giant batch of yogurt, then maybe I don't need air. That could come up in a question. Who knows? What else? What else? What else? pH probe, and that's about. I need somewhere to put the microorganisms in. There might be something else you could add on here, but microorganisms in, and then I probably need a pH probe. Why is a pH probe important? Why do we also need to monitor pH? Why is pH important? What will happen if I allow the pH to go like too acidic or too alkaline? What might happen? Yeah. So optimum pH for enzymes activity. Otherwise, we might denature the enzymes that live inside the microorganism's body, because every living thing has enzymes, and that will ruin it. You also need to know the difference between I can't remember if you do continuous and batch fermentation, but there are two ways you can do fermentation. Batch, like I said, my friend who tried to make alcohol at home in containers with wine, that's called batch fermentation. You seal the container, you let the microorganisms produce what they're going to produce, and then you take the product away. But then after, it's quite a slow process. You have to clean it all out. The bonuses are, if something goes wrong, you only lose one small batch. Continuous fermentation, which is what they do in factories and in industry, is where they continually take the waste away and add new nutrients and new microorganisms. So they just keep it going all year round, and they never have to stop and clean it out. But if you do batch, you're doing it in small amounts. Anyway, you might not do batch, and no, I don't think you don't do batch and continuous in IGCSE. So yeah, lots of sort of questions going to tie in with enzymes. Questions ask you what the paddle does. Why do we have a water jacket? To remove heat. Those type of things. What happens if you didn't get rid of the carbon dioxide? It could explode. It's also an acidic gas. It could change the pH. Yeah, you need to keep a supply of nutrients. What type of nutrients? Yeah, you got that. What type of nutrients are we flinging in to our fermenter? What would microorganisms need in order to grow and make their products? So think about what do living things need? Glucose for respiration, amino acids. The microorganisms need to make proteins so they can grow. So we need to give them amino acids so they can make their protein and they can grow and repair. And we need to give them glucose for aerobic respiration as well, or even for anaerobic respiration, so they can release energy to grow and produce our product for us. That's right. Yeah, you're right, glucose. And there might be some other things specific to that nutrient that it likes, maybe magnesium, maybe calcium ions. There might be something that organism really likes. Okay, but the main two, amino acids and glucose. You could say fats or lipids as well. But okay, any topics that anyone wants to go through, either for today's exam or Friday's exam? Any other topics? Feel free to ask about any biology topic that you would like to go over. Remember, for your IGCSE exam, anything can come up. Probably 50% of the marks will be paper 2 content only. The other 50% will be back from your paper 1. Now, it might be something where you think, what did they not test us on in paper 1? They didn't really do any Punnett squares or any inheritance, then you might say, right, that's coming up. Punnett squares are coming up in paper 2. So yeah, so we'll look at ecosystems. Let's have a little look at ecosystems and human influences on them. So one secondo. Okay. Woo. So how are you with your carbon cycle? You okay with the carbon cycle? You got a Punnett square in paper 1. Cool. So you should be cool then for Punnett squares, but you never know. So carbon cycle, you got carbon dioxide in the atmosphere. How does carbon dioxide get into living things? Do you know how carbon dioxide gets out of the atmosphere and into living things? I don't do maths, thankfully for me. Yeah. You may have biology on Friday as well. How does carbon dioxide get into living things? What's the process by which carbon dioxide goes into living things? And this is the carbon cycle, which relates to human influence on the ecosystem, because we can talk about how humans affect the carbon cycle. That's right, Nihat. It is photosynthesis. So taking any biology questions today. So photosynthesis takes carbon dioxide into the plant. What is it turned into, the carbon dioxide, when it goes into the plant? Does anyone know what carbon dioxide is turned into once it gets into a plant by photosynthesis? Yeah, so the oxygen is taken away and released as a waste gas. What happens to the carbon? This one is the carbon cycle. What happens to the carbon? It's not the carbon dioxide cycle. What happens? The carbon is turned into C6H12O6. As an example, yes, glucose. You don't have to write that, but it can also be turned into starch, can also be turned into protein, can also be turned into lots of other things. So complex organic molecules. Now, then gets into living things like humans. We eat plants, so we've got feeding going on. And what are humans going to do that releases carbon dioxide back into the atmosphere? After all, this is a cycle, and plants do this too. What's the process by which carbon is taken back to the atmosphere in the form of carbon dioxide? Does anyone know how carbon is released back to the atmosphere? There's actually 3 processes that release it. Yeah, so we've got respiration, which is done by, obviously, humans and plants. Some people forget that plants do that as well. So remember, plants also do that. Now, sometimes the plants die by death, obviously. You don't die by any other way, but organisms die. Obviously, a plant doesn't have a skull, but this is my little skull and crossbones. Nice one. Yeah, we've got decomposition. So what does the decomposition? What organisms carry out decomposition? That's the next process. So you've kind of got 4 major processes. Oh, sugar. Can't even... Yeah. Don't know why I've written those in capitals. Yeah, fungi, bacteria, good. Microorganisms. There is actually bigger things like worms and beetles that break our bodies up. They're called detritus feeders. But really, we're focused in IGCSE and GCSE biology on the microorganisms, the bacteria and fungi that are going to break our bodies down. Take the carbon. They're using the carbon to build protein and fat and starch and glycogen and molecules inside their own bodies for their own uses, and in the process they release carbon dioxide by respiration, but we call it decomposition, that process. Now, some of these dead things become fossil fuels. Obviously not in a minute, that takes millions of years. And what do humans do to fossil fuels? So here's your first influence of humans on the environment and how we're basically screwing up the environment, or a negative impact of humans on the environment. What are we releasing? So we've got another process here. We've got another process that is releasing carbon dioxide to the atmosphere. Yeah, they can do that. We do fractionation and make smaller. That's in chemistry. But in biology, fossil fuels get burnt. What do we call the burning of fuels that releases carbon dioxide? We call it combustion. So one of the biggest things that humans are doing to screw up the environment is combustion. And you have to know that for the carbon cycle in biology. That's releasing CO2. So have a look. There are 3 processes. I'm going to highlight these processes. 3 processes that put carbon dioxide into the air: respiration, decomposition, and combustion. And there's just one process that removes carbon dioxide. So humans have made the carbon cycle imbalanced by doing combustion, burning of fossil fuels. We're releasing carbon dioxide. But we have to acknowledge there's important reasons why we burn fossil fuels: to heat our homes, to keep us alive in the winter, to provide electricity and these kind of things, and to grow food and stuff. What are we doing to the trees that's reducing photosynthesis? What are we actually doing to the forests and the woodlands that's reducing photosynthesis? So we can then come off of this and talk about deforestation. And deforestation is having a big impact on the environment. Why do we do it? Why do humans do deforestation? Sounds like a pretty mean thing to do. That's right, but why do we cut the trees down? Right. Wood for fuel. Space. The main thing is space for farming food. Human population's growing very rapidly. Doesn't mean it's a bad thing, but we just need more space for food, space for towns and cities. People need somewhere to live. So grazing, yeah, that's right, for farm animals. So there are plenty of positives. If we stop chopping the forests down, there'd be no houses for people to live. We wouldn't have enough wood. Still, half of the world doesn't have central heating or gas heaters, and they use wood heaters to keep themselves warm in the winter. So if you say, right, illegal to chop trees down, anyone that does it gets a death sentence, well, then millions of people will die in the winter. So there are reasons why we do deforestation. However, we do need to know the negative impacts of deforestation. So what are the negative impacts, and why is cutting down trees bad? What is this going to do? You're right, we need wood for construction as well and for buildings. But why is it bad? What are the nasty effects of deforestation? So one of them, yeah, soil erosion. So the roots of trees bind the soil together, and the soil erosion can lead to droughts and infertile land. Soil erosion leads to infertile land. If you chop all the trees down, and you've seen pictures of places where all the ground is infertile. You can't grow anything in it. It becomes dead. Without trees, the land is dead. There's no minerals. All the minerals, all the nitrates, phosphates, magnesium, potassium, all these things that plants need, they wash away because there's no roots to hold the soil together. So that is bad. Leaching. So without trees, I'm not talking about the blood-sucking invertebrate that feed off the blood of people. I'm talking about without trees, when it rains, fertilizers are washed into lakes and rivers. So the fertilizers that we put on crops, if there's no trees about, they leach into lakes and rivers, and we'll talk about that as well. Also increases greenhouse gases. Yep, you're right. Increases the greenhouse gases. So what does that mean? Increase in global warming. So there's loads of damaging effects. You could have a really big question on this. Yeah, and it affects transpiration. So remember that trees send water by the water cycle up into the atmosphere by transpiration, which then condenses to form clouds and it rains down. If you haven't got any trees, you've lost one of your ways of, you've interfered with the water cycle by affecting transpiration. So that's a little bit on deforestation and humans' impact. There's a direct link there, our impact onto the deforestation. Yeah, also deforestation is destroying habitats. I didn't mention that, but there's plenty of birds and animals that rely on the trees, so that's destroying those habitats. Talking of greenhouse gases, can you name 4 greenhouse gases? A genus. What is a genus? Yeah, a good methane, CFC, water, carbon dioxide. Brilliant. There's 4 right there. Well done. So, someone's asked, what is a genus or genus? So a genus is a taxon. What's a taxon? It's one of the ways that we classify organisms, and it's just above species. So our genus is Homo. There are no other homos on the earth other than humans. We are Homo sapien. Our species is sapien. If you are the same species, you can sexually reproduce and have fertile children that can go on and reproduce. If you have a tiger and a lion, they may be of the same family, but they're not the same genus. This means they can have sex and have children. They have a liger as a child, but that is an infertile hybrid. There are no baby ligers. Well, there are, but the liger cannot go and reproduce with another liger. So if you're not the same species, you can't reproduce. So that's how we define a species. Species can produce fertile offspring. But the next closest group we can put ourselves into is Homo. So we have something called a binomial naming system. Let me just run through the old binomial naming system. There's a binomial naming system, and we write it like this: Homo sapien. Now, Homo erectus, or another Homo that used to live on the earth, was of the same genus as us, was called Homo habilis. We have its fossils. We could have attempted, if we could go back in time, you could go back in time and marry a Homo habilis and have sexual intercourse with them, but you wouldn't produce fertile children because you're not the same species. So a genus is another group. There are 7 groups, and you might know the rhyme to remember it. Kings play cards on fat girls' stomachs. Some people prefer king prawn curry or fat greasy sausages. Some people prefer that as a mnemonic, and what that stands for is the 7 levels of classification, which is kingdom, phylum, class, order, family, genus, species. Now, so if I said to you, what kingdom are you in? You are part of the animal kingdom. What phylum are you in? You're a vertebrate. You have a backbone. So we actually share a we share a commonality with every animal. You're grouped in animals with insects and beetles and worms and jellyfish and apes and tigers. And then we'd say, okay, let's separate and make it a little bit more defined. Right, you are a vertebrate, you have a spine. And then we'd say, what about your class? Your class is mammalia, you're warm-blooded, hairy, and you give birth to live young. So we still have some members of our class with loads of mammals on the earth. And then you go down to your order, which is, I don't know what our order is, and then carnivora, we're carnivores, we eat meat or something like that. And then family, I don't know, we're primates, like, so, and that's as close as it gets. We have no one in our genus left, but we still have primates like monkeys, orangutans, gorillas, chimpanzees. They might be in the same family as us. So genus is just a level of classification. And as you go down, you become more closely related. Yeah. And when you write this, we tend to write this all in lowercase. So I shouldn't have put a capital S there. And this is known as, now instead of naming a human would be really hard work. What do I mean by the binomial system? A biologist will name every animal by the last 2 classification levels or taxons. They will say their genus and species. So a great white shark is Carcharodon, Carcharinus, or Carcharius, something like that. Carcharodon, Carcharinus, or something like that. So you name the last 2, and then someone goes, oh, you don't go, hi, my name's Tom, I'm an animal, I'm a vertebrate, I'm a mammal. You go all the way down to saying I'm a sapien. You just say I'm Homo sapiens. So binomial means 2 names. We give every animal or plant or bacteria or organism 2 names, their genus and their species. We start with genus, start with species. So if I did this and I write Xena sapiens, you might say, oh, who's the most closely related? These 2 are. They share the same genus. They're more closely related. This one might sound similar, but it doesn't have the same genus. So you can see who's more closely related. Okay. Mycoprotein. Mycoprotein is like protein made from fungus. That's in genetic engineering. So protein made from fungus. You can make mycoprotein in a fermenter. You can grow the fungi in a fermenter, and they produce like the mycoprotein, or is actually, sorry, mycoprotein is single-celled protein, isn't it? It's actually, you're eating the fungus. So when you eat like Quorn, the vegetarian sausages that make the Quorn, they use a fungus that you're eating, which is a single-cell protein. The benefit of it is very easily digestible. There's no bones, no skin, no ligaments, no tendons. When you eat chicken, there's tendons, ligaments, and skin to digest. Mycoprotein is very digestible, but yeah, it's all single-celled, so the texture is soft and mushy. People are like, it's not really like eating meat, it's all mushy. And that's the unfortunate thing with mycoprotein, is that the texture, some people don't like it. It's soft like marshmallow because it doesn't have tendons, ligaments, bones and skin and those kind of things that you get in meat. There's no tissues all being joined together. Okay. Has anyone got eutrophication in their exam system? Are any of you guys doing eutrophication? Do any of you have to do eutrophication? My God, that's the worst bit of water I've ever seen. Yeah, so if you have your crops, and I have my farmer over here, Mr. Farmer or Mrs. Farmer Giles, or whatever her name is. So she's farming, and she's putting fertilizer on her crops like this because she wants them to grow really well. She might be feeding them nitrates. Nitrates allow the plants to make amino acid and proteins. Proteins are needed for growth, so a very important fertilizer to add to your plants. The problem is that when it rains, what's going to happen to these fertilizers when it rains? Does anyone know what happens to the fertilizers when it rains? So this is about water pollution. It's also in AQA Combined Science. We have a process called leaching. Fertilizers wash into rivers and lakes and pollute the water. Okay, and that is called leaching. Now sewage also does this. So some companies dump their raw sewage into the seas and the rivers. Sewage also does this. What happens is the fertilizers go in here. Now over here in our lovely lake, yeah, very good. So over here in our lovely lake are plants called producers. And producers give out oxygen. Plants do photosynthesis and they give out oxygen. What do they require for that? They require sunlight and they give out oxygen. However, as Nihat said, we get an algal bloom because of the fertilizers. We get algae bloom or grow all over the top. They block the sunlight. Our producers die without sunlight and they no longer produce oxygen, and then our fish die in the lake. Also, you can talk about decomposers, microorganisms, feed. So there's two ways the oxygen gets used up because there's no plants. Decomposers use up also. Decomposers use up the O2 in decomposition by doing respiration as they feed on dead algae. And this whole process is called eutrophication. It could come up as like a 5 or 6 marker if you're doing biology GCSE or biology IGCSE. If you're doing combined science, you only really need to know this bit. And why can sewage do this? When you go for a poo or a wee, your feces and urine contain nitrogen, lots of nitrogen, because we eat lots of meat. And then if that washes down into the water, or we eat lots of protein, I shouldn't just say meat, we eat lots of protein. And then if that waste washes down into the water and the sea, it means the algae feed on those nitrates and they grow and they block the sunlight and kill the plants that oxygenate the water, the producers. They stop photosynthesis. And that ends up killing the fish and toxifying the whole lake. So that's eutrophication. Is there any other topics? What are the different type of bacteria like nitrogen fixing? Oh yeah, so are you doing the nitrogen cycle? So it depends whether you're doing the nitrogen cycle in your exam board or not. Okie dokie. Nitrogen cycle is a lot harder than the other cycles, but essentially you've got nitrogen, and nitrogen can go by 2 ways into the soil. So there's 2 types of bacteria here. We got nitrogen fixing bacteria, and these ones in the soil, these guys just live free in the soil. And then we have lightning can fix nitrogen into the soil as nitrates. Yeah, you've got nitrogen and denitrifying. You've got nitrifying and denitrifying bacteria. Essentially, if I just draw a person, when people die and plants die, again, you've got a bit of death and decay going on here. So when people and plants die, there are bacteria that feed on us. We become a substance called ammonia, and you might remember ammonia from chemistry has the formula NH3. So you might remember chemistry has the formula NH3. I don't know if you need to know about ammonium ions. Oh yeah, you do. So it's a bit long. I've not left myself any room whatsoever here, which is a bit of a bummer. So the ammonia becomes, if you've done your chemistry, ammonium ions, which are NH4 plus. These then get nitrified or oxidized into nitrites, NO2, and then are further oxidized into nitrates, which are NO3. Once you get into a nitrate form, you're ready to be sucked into the roots of plants through the water and through active transport. Remember, these go, I haven't done a very good job here with my nitrogen cycle, but that's what happens when you're just doing it off the cuff. Nitrates can go straight into the roots like that. Now, there are also, in certain plants, certainly not trees, there are nitrogen fixing bacteria living in the root nodules. So you've also got nitrogen yeah, that's right, root nodules. You've got nitrogen fixing bacteria living in the root nodules, and they're called leguminous plants. So if you're vegetarian, any vegetarians, baked beans, peas, kidney beans, lentils, chickpeas, all of these kind of beans and peas are very high in protein. The reason is those plants have nitrogen fixing bacteria in their roots. They take nitrogen from the air, add it to oxygen, and form nitrates, which can go straight and be used by the plants. It's called a symbiotic relationship. The plant's roots give shelter to the microorganisms and stop it being eaten by its predators, and the microorganisms supply the plant in return as a deal with nitrates for making proteins. However, other bacteria in the soil, called nitrifying bacteria, turn the ammonia into nitrates. And then we have the one bacteria I haven't put here is denitrifying bacteria. There are some bacteria that which I've not left any space for doing. There are some bacteria called denitrifying bacteria that just chuck. Oh yeah, I've not left myself any space. Pretty bad. Denitrifying bacteria. These take the nitrates, NO3. They steal the oxygen, leaving behind the N, and they chuck that back in the air as nitrogen. They take the oxygen for respiration and throw the nitrogen into the air. So if you were doing chemistry, that is a reduction. They're removing oxygen. That's what denitrifying bacteria do. Whereas nitrifying bacteria add oxygen to the ammonia to make it go to NO2 nitrite and then NO3 nitrate. So nitrifying bacteria oxidize, denitrifying bacteria do reduction by removing oxygen, and they chuck the nitrogen back in the air. Yeah, and nitrogen's needed by all living things to make amino acids, and that's the nitrogen cycle roughly. So your 4 types or 5 types of bacteria are free living nitrogen fixing bacteria that just fix nitrogen into the soil as nitrate, and they live just free. Then you've got nitrogen fixing bacteria living in plant roots. Then you've got nitrifying bacteria that feed on dead organisms and turn the ammonia and ammonium ions into nitrates, which can then be sucked up. And then you have denitrifying bacteria that take nitrates and turn them straight back to nitrogen in the air. Yeah, so lightning, you got N2, you got nitrogen and oxygen, 78% nitrogen in the air. What is it, 28 or 29? I can't even remember. 21% oxygen in the air, 78% nitrogen. When lightning cuts through the air, there is so much energy that it is able to break the very strong covalent bond in the N2 and the very strong covalent bond in the O2. So it's enough energy in the lightning to break the bonds, activation energy, and force the nitrogen and oxygen together to make nitrate. So lightning can just make nitrates like that, join the nitrogen and oxygen. It also, I think it also forms ozone, which is like O3. They say you can smell it when lightning's been in the air or something like that. But yeah, so it's the incredible high temp. I don't think you would need, yeah, there's so much energy that it causes the nitrogen and nitrates and oxygen to react. Do you know about acid rain, guys? So you need to know that some fossil fuels, such as coal in power stations, contain sulfur. When we burn them, forms S plus O2, makes SO2, forms H2SO4, sulfuric acid in the clouds, rains down as acid rain. This changes the pH of soil, kills the trees, the pH of water, kills the fish and marine life, organisms in the water, and erodes buildings and stuff, and that's acid rain. I'm going to have to shoot off in a min. Any questions? Why do plants need nitrates? Lloyd. So plants need nitrates, as Nihat said, to make amino acids. So amino acids, okay, contain nitrogen. Don't ask me why I drew a circle around it. But this is the formula of an amino acid. And amino acids have carbon and hydrogen. Plants can get carbon and hydrogen. Where do they get that from? Carbon dioxide, H2O in photosynthesis. So they're cool with that. But they need to make amino acids. Proteins are made of this unit, which is called an amino acid, and they're made of lots of these. They do have slightly different formulas, but they have nitrogen in them. And so plants need nitrates to make amino acids. How do we get our amino acids? That's simple. We wait for the plants to make their the plants do the difficult bit. They suck up the nitrate, they split it apart, they join it with the glucose, and they turn that into an amino acid, and then they bind them together to make protein. We eat the plant, we break down its protein into amino acids, then we rearrange them in a different order and make our own proteins, which is why we look different than plants, because we're made of different proteins, but we've still got the same amino acid building blocks. Yeah, they have the carboxyl functional group there. Yeah, and hence the name. This is the amine, and this is the carboxylic acid. So the name is amine, which is an alkali, and acid. This is, so it's like alkali and acid. They're a bit of both. Amino acid, although, yeah, don't want to go too much into that. Yeah, there's an amine group, NH2 on one end, and then COOH on the other end. Lloyd, we don't need to take in nitrates. We don't need to have nitrates, no. We just get our nitrogen by eating either other animals or plants. So animals are cheats. We wait for the plants to produce the protein for us, then we eat them. And we use their proteins. We can't do that. So without plants, we're all dead. Without the sun, we're all dead. It doesn't matter if, I can live on... Yeah, without plants, we can't get protein, because we don't have a way of taking nitrogen out the air and into our body to make protein. We can't do that. In fact, we can't make glucose, we can't make carbohydrates. Plants can survive without us. We can't survive without plants. So the earth can survive without animals, but it can't survive without animals can't survive without plants, essentially. I know people say, oh yeah, but we give plants the oxygen. There's other ways for them to do that. Or we give plants the carbon dioxide. There's enough carbon on the earth for them to live without us. Right. You do need to know why. You also need to know why pyramids are shaped like this. And I don't mean the pyramids in Egypt. I mean pyramids of biomass or pyramids of energy. So why do pyramids of energy get smaller with each energy level or trophic level? So yeah, big to small in biomass or in pyramids of energy. They have to go big to small. Yeah, there are other ions plants need, by the way, Lloyd. There is magnesium needed to make the leaves green, make chlorophyll, which absorbs sunlight. So a plant low on magnesium will have yellow leaves. There's nitrates needed by plants to make them grow. So a plant without nitrates will be short and stunted. There is potassium and phosphates as well. Phosphates are needed to make DNA, and potassium is also needed in a plant. And the fertilizers are called NPK fertilizers for their elemental symbols. N for nitrogen, P for phosphates or phosphorus, and K for potassium. Yeah, that's right, Nihat. So not all the energy is passed on from one level to the next. About 10% passed on. However, if it's in the sea, more get passed on from the producers, because remember, when a goat eats a plant, it probably won't get the roots, and it probably won't digest it all. But if you're eating algae have no tissue. They're just single-celled organisms, just a big green gloop. You're going to be able to digest that and get lots of energy. So in the sea, we will find, this is about as far as you can get on land, but we'll find in the sea food chains which have this is the worst thing, but they can go on and on. You maybe get 7 steps in the sea, and it's all because maybe 20% of the energy from the algae gets passed to the primary consumer, and then from there it's 10%. The sea is more efficient. There's a lot less energy wastage. I guess the water acts as like less heat loss to the environment and stuff. But yeah, so why does the energy levels get smaller with each step? This is because of heat losses. Animals that need to keep warm, like mammals, have to make heat. Respiration, movement, running around, chasing, hunting, prey, or if you're prey, like a rabbit trying to escape a fox, you're going to run a lot. So none of that energy can go on the body. It's all lost to the environment. And excretion, breathing out carbon dioxide. Carbon dioxide has mass, so if it's a pyramid of biomass, it's going to decrease when you breathe out carbon dioxide. Also urinating and feces, and anything you eat might be undigestible parts of it. So not all energy is passed on. In fact, this is the scientific argument for being a vegan. So the scientific argument for being a vegan or a vegetarian, they say, well, look, here is the human, and we're eating the chickens, right? There wouldn't be that many levels to the chicken. There'd be 2 levels. So they say, here's the human right here, and you eat your lovely chicken burger. But that chicken burger has needed all of this plant material to feed it. If we skipped eating the chicken and you ate the calories from plants, you would only need that much plants, and you could actually feed another 10 humans. So if we didn't eat the chicken and we ate the corn the chicken ate instead, you could feed 10 humans on the amount of corn that one chicken ate. So one chicken ate loads of corn, we eat the chicken, the chicken only got a tenth of the corn, and then we only get a tenth of the chicken on us. So some people say, look, if we skip the chicken and go straight to the corn, we can feed humans with all that corn, and instead of wasting it. So so that's the reason some people say they're vegans or vegetarians. They go down the food security, the route of food security. Other people just say, look, it's really cruel to eat animals, and in this modern day and age, we can get our nutrients. They say, we understand in the past that eating animals you needed to for protein, but now there's so many alternatives than eating animals and farming animals. And they point to farming being a really bad practice, deforestation, global warming, and they sort of say, look, that's all contributing it. My theory is if you went down everybody being vegans, you may solve one issue and you probably create another issue that we're unaware of at the moment, because not everyone's a vegan. A bit like driving electric cars. Everyone thinks driving electric cars is good for the environment. If everyone drove a Tesla, the batteries in the Tesla contain precious metals or toxic metals like lithium and things like that. So we might go from one problem to another problem and find that if everyone's driving those, we destroy the world that way. So you're never really going to get, you know, you can't really preach to people that you're being super green because you don't know the consequences. Potentially there could be problems if everyone's vegan. Who knows? They're unforeseen at the moment because we haven't had to sit and think about them at all. But potentially there could be a problem if everyone became vegan. But one of the arguments for veganism is that if you don't eat the chicken and you eat the animal in the lower level. One of the arguments against being vegan or vegetarian is that we, as mammals and omnivores, we have evolved to eat meat, and plant material takes a long time to digest and it's quite inefficient eating plant material. This is great for losing weight, but if you go on a vegan diet, you're going to lose tons and tons of weight. But we might find there's a massive problem with malnutrition. This is just one suggestion. This is definitely not true, but it could be. We might find there's a problem with malnutrition if everyone becomes vegan, because when you eat plant material, a lot of that material is impossible for you to digest, and it's not processed. If it's not processed, it's difficult to digest, and you spend a lot of energy digesting. So if you eat a McDonald's cheeseburger and it's 1,000 calories, 800 calories might go on your body and 200 calories in the enzymes and the heat and everything to chop it up. If you eat 1,000 calories of celery, you might spend 900 calories chopping it up, and only 100 calories, or joules, sorry, should use joules, goes onto your body. So different foods take different amounts of energy to digest, and no one really takes account of that. They just look at the numbers on the back of the packet. But yeah, anyway. GM foods, it depends what you mean by are they safe to consume. Absolutely. Essentially, it's just protein, carbohydrates and fats. So there's nothing in a GM food that's going to be dangerous for your body just because it's been genetically modified. You're going to digest that and destroy that in the digestion. However, you may say ethically, are GM foods right? That's a different argument. Are ethically GM foods are safe to consume, but are ethically GM foods right? Genetically modified foods. Some people would say if I don't want to eat GM tomatoes, right? I don't want to eat genetically modified tomatoes. I don't want to eat a tomato that's got a gene from a carrot in it. I want to know my tomato is my tomato. So this is not me. This is me being someone else who's really ethical about it and says, I don't want no carrot in my tomato. I want to know I'm just eating that. However, if the organic farmer down the road is growing his organic carrots and he or she says, buy my carrots or buy my tomatoes, they're organic, how does he know that a bumblebee hasn't gone from the GM farmer's field into his field and brought over the crossed GM genes via pollen and eggs? So all of this, once you start doing GM food, all the food in the world becomes GM, because we can't say to a bumblebee, you're not allowed in that field, that's organic, mate. They're not using genetically modified. That bumblebee is carrying the genes from the pollen, like the sex cells, into the next field. So yeah, it's more ethical with GM foods. Once that you get contamination with wild plants as well. Like, we don't know if we put a gene from a carrot in a tomato and then that tomato plant, I don't know, fertilizes with a wild plant that's not a tomato, but you get cross species. Those genes then get into the wild. We don't know. That might create problems further down the future we can't envisage. You know, we can't envisage these problems. Maybe we're going to let some genes loose in the wild that are going to get taken in by another plant that's going to get an advantage over all of its competitors and wipe them out and make them extinct, all because we get put out this gene which wasn't supposed to end up in that particular plant. Who knows? But yeah, it's mind-boggling to think of all of these things. Listen, guys, I've got to shoot now. I've got a meeting at 1. Good luck good luck with all your exams this week. On Thursday evening, I'm doing a biology revision lesson as well. I'll put loads of exam questions up and go through them. All right, guys, take care and yeah, enjoy the rest of your day. Bye, guys. Don't forget to hit the notification. And that if you hit notify me, then you'll be notified of any live streams I do. Over the summer holidays, I'm still going to be doing live streams to prepare people if they're taking A-levels for chemistry and biology. So, all right, guys, take care. Bye-bye.